Hi everyone, I'm Dr. Preeti. I hope all of you have attempted the recent MCQs that I posted on MicroRNA on my Telegram and Facebook group. And here is a very short video discussion about microRNA, which is now a very important topic for your upcoming exams. So before we begin with the synthesis and the disease association, we must first get back to our basics. What was our basic? DNA by transcription forms RNA, and RNA by translation forms a protein. So see over here, the RNA which is coding for a protein is known as a coding RNA, right? It's a coding RNA. But if an RNA does not code for a protein, like you see out here, if an RNA does not code for a protein, it is a non-coding RNA. And that is what microRNA is. MicroRNA is a non-coding RNA. Micro because only 22 nucleotides, roughly 22 nucleotides, and it is a single-stranded RNA. What does it do? Please note, this is your previous year question. This results in post-transcriptional modification. It causes post-transcriptional modification. So please remember, micro because 22 nucleotides. It is a single-stranded RNA and it causes post-transcriptional modification. So now we are going to first understand the synthesis of microRNA, and then we are going to talk about the diseases. Before I do that, let me tell you that talking about the synthesis, it's going to happen in the nucleus first. So do you see? The DNA by transcription will form primary microRNA, multiple loops, hairpin loops, primary microRNA. One of these loops will be cut out by DROSHA and DGCR8. What do we have? Next we have pre-microRNA, precursor microRNA. So DROSHA and DGCR8 have brought out one hairpin loop. This hairpin loop is now going to move out of the nucleus via a port, exportin 5. See, it has moved out via exportin 5. So where has it come? It has come out into the cytoplasm. The precursor microRNA is out in the cytoplasm, and now comes a molecule which is going to dice it. So it's a hairpin loop structure. I want to get rid of that loop. So I bring dicer. Dicer comes and gets rid of that loop. So now the loop is separate. As you can see, the loop is separate and a double-stranded duplet is in front of me. MicroRNA, duplet. But do I want double-stranded or single-stranded? Exactly, we wanted single-stranded microRNA. So here we have one, and that goes and sits in a complex known as RISC. What is RISC? Here I have a complex of mi-RISC. What is this complex? So let's study about it. It is RNA-induced silencing complex. Look at it. It's going to go and sit on the particular strand where protein synthesis had to happen. So is it letting the protein synthesis happen? Do you see? No, it's sitting like a roadblock. It's not letting protein synthesis happen. It is RNA-induced silencing complex. See, it's a roadblock, no protein synthesis. RNA-induced silencing complex, and that is exactly how I say that my mission is accomplished. miRNA has formed a RISC complex, mi-RISC complex, and that is not letting the protein synthesis occur. So can I say that microRNA is a non-coding RNA, and via which molecule? They ask you this question, what is the molecule via which it silences the protein production? The name of the molecule is RISC. I repeat, RNA-induced silencing complex. So microRNA is formed. Now we need to know its association with the diseases. So see, we have two microRNAs. One is a tumor suppressor microRNA in our body, which is a good one, which is suppressing the tumors. And the other are the oncomers, which are the dangerous ones. They can cause cancers. So one are good people and one are bad people. Let me give you an example of the good people first. Tumor suppressor microRNAs. Examples of these are microRNA 15 and 16. And if something goes wrong in these microRNA 15 and 16, if they get deleted, please remember the patient lands up with a disease that is CLL, chronic lymphocytic leukemia. I hope everyone remembers this from HIMAT. MicroRNA 15, 16, the good people get deleted, person lands up with CLL. What about the bad people, the oncomers? Oncomers, the numbers that you have to remember for oncomers are miRNA 155 and 200. So please remember, oncomers are your bad people. if they increase in your body, it's going to be dangerous. It will result in cancer. So microRNA 155 and 200, these are your bad people, the oncomers, cancer causing, and if their level increases, patient lands up with B cell lymphomas. So these are the two disease correlations of microRNA that all of you have to know. Having said that, there's another disease correlation that you've got to know, and that is to do with one of the proteins which was dicing the hairpin loop. Do you remember that? The dicer, yes. So yes, that's a very important gyne question that is now put up, that what is the disease that happens if there is a dicer mutation? So let me take you back. Dicer is something that is going to basically be one of the processing proteins, right? And if this undergoes any mutation, the patient is going to land up with ovarian and testicular tumors, particularly the sex cord stromal tumors, right? So if there's any mutation, patient lands up with the sex cord stromal tumors. So that was about it with respect to the disease part of it. Everyone, please do revise the synthesis and the diseases. Thanks for listening this short video. Good luck.